Question 5. Have I been lied to about the origin of life? A recent survey asked people if they thought the following statement was true or false. Scientists researching the origin of life have created simple life forms from scratch. That is, in the laboratory experiments that approximate Earth's early atmosphere, scientists have mixed chemicals believed to exist before the first life forms to successfully create simple life forms such as bacteria. 73% of participants believed that statement to be true. In reality, this couldn't be further from the truth. Why have so many people been misled? And what does the evidence actually say? 4 of life's biggest questions include origin, meaning, morality, and destiny. The origin of life question will help answer where we came from and if there's any inherent meaning to life. In episode number 1, we discussed big picture cosmic origins. Now we'll discuss the beginning of life itself. So, what exactly is life? A basic definition is an organismic state characterized by capacity for metabolism, growth, reaction to stimuli, and reproduction. A more technical definition says it's a process that takes place in highly organized organic structures and is characterized by being pre-programmed, interactive, adaptive, and evolutionary. Origin of life research attempts to discover how life could have arrived from non-living material on the early prebiotic Earth. Many people assume that the problem has been solved due to the generalized idea of evolution. Darwin's initial research for his theory took place almost 200 years ago. He suggested that all living things were related and that they could trace their lineage back to a last universal common ancestor. But that begs the question, where did the first living thing come from? How did it begin? The International Society for the Study of the Origin of Life says when Charles Darwin published The Origin of Species 150 years ago, he consciously avoided discussing the origin of life. The basics of evolution teach that if you have variance among a living population, and those populations reproduce based on their ability to survive, you get natural selection. Well, there's a few assumptions built into the equation. Variance assumes that you have various forms of life. Reproduction simply assumes the ability to reproduce. Neither of these things exist on a prebiotic Earth. So the question is, how do we get life without presupposing, well, life? All living cells are immensely complicated due to their various moving parts, such as these. A cell can be thought of like a laptop computer. It's made up of many different individual components. But any one of those individual components is also made up of smaller parts that allow it to function. But even these smaller pieces have individual building blocks of their own. To make matters even more complicated, as it is with the cell, you can have all the parts present and fully assembled, but they're entirely useless without some type of specifically sequenced code or information. For now, we'll just say it is extremely complicated. So we've got 2 big problems on our hands. First, you need the building blocks of life. The 4 fundamental building blocks of a living cell include carbohydrates, lipids, proteins, and nucleotides. Dr. James Tour is a synthetic chemist with over 750 research publications, over 130 granted patents, and he's considered among the top 50 most influential scientists in the world today. Discussing conditions presumed to be on a prebiotic Earth, he has this to say. When one looks at a typical textbook, one will see some prehistoric pond and molecules, and those molecules coming together and forming a cell, and those cells coming together and some slithering creature coming out of this pond. That is fallacious. There is no truth in that. These little pictures of molecules coming together to form the first cell are fallacious and ridiculous. The origin of life community has not been honest. He also says, it's very simple. 4 classes of compounds. You have to make them from what's available on a presumed prebiotic Earth. We don't know how to build the molecules. time is actually the enemy. You let these chemicals that have been made sit around, they show the degradation in a period of weeks. Weeks is the twinkling of an eye when it comes to prebiotic timescales. The chemicals decompose, so to think that the molecules could be made and sit there waiting for other molecules to come in, it doesn't happen. Organic chemistry doesn't work that way. For more information on the building blocks problem, check out these resources. Problem number 2, where did the information come from? We often take for granted some of the greatest scientific achievements from recent history. People like Charles Darwin had no idea what DNA was or how it works. So, how does it actually work? Avid proponent of atheism, Richard Dawkins, has this to say. The genetic code is not a binary as in computers, but a quaternary code with 4 symbols. The machine code of the genes is uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be interchanged with those of a computer engineering journal. Take, for example, this set of 1s and 0s. To most people, it might not mean much, but in computer code, it says you're watching QUEST Questions. Now, computers only have 1s or 0s, but cells have double the options among the 4 nucleotide bases. So we might ask, maybe the DNA sequence came about by purely natural processes. This is where the chemistry gets interesting. If you were to unwind a strand of DNA, you would essentially find the backbone and the code. The type of bond that attaches the code to the backbone is completely impartial. It doesn't prefer any one of the 4 characters of code over another. In other words, it is completely random. This makes some of the math going forward pretty easy. What are the odds of just 10 bases falling into a specific sequence? Exponential math tells us it'll be 4 times 4 times 4 times 4 and so on, or 4 to the 10th. And what does this come out to? About 1 chance in 1,048,576. And that's just 10 bases. Dr. Tor goes on to explain how origin of life research can mislead the public. They tend to manufacture chemicals at unnatural, prebiotically irrelevant concentrations. They mix these chemicals in a specific order under precisely controlled, unnatural laboratory conditions. They end up producing mixtures that contain small, unusable concentrations of the desired building blocks of life. Lastly, they write a paper with unjustified extrapolations. Take this one, for example, which says, researchers say they created a synthetic cell. But what did they actually do in the laboratory? Well, they took a pre-existing cell, removed the DNA code from the cell, made an exact copy of that DNA, and then stuck it into another pre-existing, fully assembled cell. That's like taking a computer, making a copy of the code, downloading it to another computer, and then saying you made an all-natural laptop. The DNA they stole from the first cell contained 582,000 specifically sequenced characters of genetic code. If you thought it was hard to get the sequence of just 10 bases, imagine 582,000. So your next question might be, maybe the DNA code used to be much simpler. Well, scientists have looked into that too. A recent study shows that the theoretical minimal cell still must have 530,000 base pairs of nucleotides. Maybe we're still thinking too big. Doug Axe is a professor of molecular biology. He wanted to find out what the odds are of a random gene potentially producing any cellular function. He found that a random sequence of an average length gene had unimaginably low odds of producing a function. How low, you might ask? About 1 chance in 10 to the 77th power. How big is 10 with 77 zeros after it? For perspective, estimates show that the number of all the grains of sand on all the beaches and all the deserts on the entire Earth come out to a measly 10 to the 21st. Needless to say, the odds are extremely low. Francis Crick made the historic discovery of the DNA double helix in 1953. Just 4 years later, he also was the first to propose how the DNA code works. Understanding the implications of his discoveries, while also holding true to his atheistic worldview, he was compelled to present the directed panspermia theory in 1972. One of the greatest minds of the 20th century, proposing the theory that organisms were deliberately transmitted to the Earth by intelligent beings on another planet. 4 decades ago, Sir Fred Hoyle calculated that the odds of all the proteins coming about by chance to form a simple cell is about 1 in 10 to the 40,000th. 3 decades of research later, and the numbers remain true. The transformation of an ensemble of appropriately chosen biological monomers into a primitive living cell capable of further evolution appears to require overcoming an information hurdle of super astronomical proportions. An event that could not have happened within the time frame of the Earth, except, we believe, as a miracle.